Hello everyone. Today we are going to continue with biology chapter number 4, molecular basis of inheritance. In this topic, in our last lecture, we have covered the topic introduction of translation and the requirements of translation. We have studied about the requirements of translation in detail and we have studied about the definition of translation. So today we are going to study the mechanism of translation. Okay, that is stages of translation. There are 3 stages in translation: initiation, elongation and termination. Before starting, let us revise the definition of translation. It is the process in which the messenger RNA is generated by transcription, is interpreted by the ribosomes to generate a particular amino acid succession or polypeptide that will fold into an active protein. In short, I will tell you the definition, easy definition. Okay, translation means it is the mechanism in which the codons of mRNA are translated and specific amino acid in a sequence form of a polypeptide on ribosomes. Okay, so translation is what? It is the mechanism in which codons of mRNA are translated and specific amino acid in a sequence of form of a polypeptide on a ribosomes. Okay, so let us start, and we have studied about the requirements in detail. Okay, that is amino acids, DNA, RNA, ribosomes. And we have studied the 3 sites of ribosomes: P site, A site, E site. Okay, so today we are going to study further, that is mechanism of translation. In mechanism of translation, there are 3 steps: initiation, elongation, termination. All these steps we are going to study one by one. So starting with the first step, that is initiation. In initiation, the first and essential step is tRNA activation. It is the most essential step, okay, in initiation. Before translation, we have to do the activation of amino acids. It is an essential step before translation, which initiates, for which ATP is essential. Okay, let us see the reaction of activation of amino acid. Amino acid plus tRNA plus ATP. In the presence of Mg+2 ions, it is activated and it gets converted into amino acyl tRNA and AMP is formed. Okay, and this amino acyl tRNA is further used in the process of translation. Okay, amino acyl tRNA is further used in the process of translation. Moving on to the second step of initiation, that is small subunit of ribosome binds to the mRNA at 5' end. Okay, there are 2 subunits we have studied. In that, the smaller subunit of ribosomes attaches at the 5' end. Okay, of mRNA. Okay, this is binding to the mRNA at 5' end. You can see here 5' end. Okay, so then initiator codon AUG is present at 5' end. We have studied that. Okay, what is the codon that is present? Initiator codon AUG is present at the 5' end, which initiates the process of translation. And then initiator tRNA binds with initiation codon AUG by its anticodon UAC. Okay, and for this process there is a initiation factor which helps them. Okay, initiation factor helps them for the process of initiation. Then step number 3 in initiation is once the ribosome binds. Okay, once the ribosome binds with the initiation codon, then what is happening? Once ribosome small unit reaches AUG, initiator tRNA attaches to the AUG and by its anticodon UAC. there is a bonding. This bond, they are attached to each other with the help of a bonding, that is known as hydrogen bonds. So hydrogen bonds form between the mRNA codon and tRNA anticodon. Okay, you can see here in this diagram hydrogen bonding. Okay, AUG and UAC. AUG is of mRNA and UAC is of tRNA. Okay, so codon and anticodon pairing takes place with the help of hydrogen bonds. It carries activated amino acid methionine. Okay, what is happening here? The starting amino acid in prokaryotes. Here the cases are different. In prokaryotes, the starting amino acid is N-formyl methionine. Okay, N-formyl methionine, and in eukaryotes it is methionine. Okay, in prokaryotes, N-formyl methionine is the initiation codon, starting amino acid, and in eukaryotes, regular methionine is the initiator. Okay, starting amino acid. Now, what happens? The large subunit of ribosomes join with the smaller subunit with the help of Mg+2 ions. You can see here the large subunit and smaller subunit both have joined to each other, both have assembled with each other. Okay, both join with each other, and Mg+2 ion plays the important role here of joining these two ribosomes. Now, initiator charged tRNA with activated amino acid, that is methionine. Okay, that here activated amino acid is methionine. You can see here in this diagram, is occupying the P site of the ribosome. You can see here the P site of the ribosome. Okay, so final position of initiator tRNA is P site and A site is left vacant. Okay, the first amino acid tRNA complex enters at P site. Okay, first amino acid complex enters at P site, and then the remaining amino acid complex are going to enter from A site. Okay, that I have explained you initially in the last lecture. So, next step is elongation. Okay, next step is elongation. In elongation, the first step is codon recognition. Okay, what is the first step? Codon recognition. The incoming amino acyl tRNA, you can see here amino acyl tRNA. Okay, amino acyl tRNA comes here one by one. First amino acyl have entered at P site. Then later on all the amino acids are going to enter at A site. So, incoming amino acyl tRNA to A site, there is a hydrogen bond formed between the mRNA codon and tRNA anticodon. Here energy is required. There is recognition. First time what is going to happen? Codon recognition is going to take place. Amino acyl tRNA enters at the ribosome from A site. Anticodon binds with the codon by hydrogen bond. Is it clear? I repeat. In elongation, first step is codon recognition. The amino acyl tRNA complex enters at A site, and then the codons are read, and as per the codon, the anticodon hydrogen bonds are formed between codon and anticodon. Next, in elongation, second step is peptide bond formation. Okay, peptide bond formation. See here what is happening. Ribosome catalyzes the formation of a peptide bond. Okay, you can see here the A site and P site. What is happening? The amino acyl complex, they are moving. Okay, this mRNA is moving actually. mRNA keeps on moving, and when it comes to here at the P site, what is going to happen? The peptide chain is going to form. Peptide chain is longer by one amino acid. Okay, polypeptide chain is transferred to tRNA at the A site. This polypeptide chain gets transferred to A site. This is moving and moving. The peptide chain moves to A site. You can see here the movement. Initially we can see here at the P site. The chain is formed at the P site, and then it moves at the A site. Okay. And one by one, all the amino acids are joined. In this way, a polypeptide chain is formed. And here, enzyme ribosomes act as a catalyst. You can see here, ribosome catalyzes the formation of a peptide chain. So, who is involved here? The enzyme ribozyme. They are acting as a catalyst. That means they speed up this process. Okay, catalyst means they speed up the process of elongation, that is peptide chain formation. At this time, first tRNA at P site is kicked off. Now, what is going to happen? The first tRNA that have entered already, when the peptide chain moves to A site, at the same time, the tRNA moves to E site. E site means exit site. So, from here, E site, the chain is kicked off. Okay. You can see here in this step, third step, okay, translocation. Third step is called as translocation. Translocation also requires energy. Okay, ribosomes move. Who is moving? I will define the translocation. The tRNA at A site carrying a dipeptide at A site. Okay, dipeptide at A site moves to the P site. It is moving to the P site. This process is called translocation. Okay. Now A site is empty. Okay. When the tRNA from P site, what is going to happen? The empty tRNA is going to move to E site and it will exit from E site. So tRNA from A site to P site, polypeptide return to P site and ready for the next polymerization. Okay. A site is now empty. And next amino acid tRNA can bind at A site. So this process goes on repeating. Enters at A site. Okay, enters at A site. Chain is formed. Empty tRNA moves to E site and it gets exit. Okay. Next and the last step of translation is termination. Okay. Now here termination means to stop the process. Termination of the process. Okay. Termination means stop. Okay. And before that, all the events like arrival of tRNA, okay, arrival of tRNA, amino acid complex, formation of polypeptide chain, ribosomal translocation and removal of previous tRNA are repeated. This process goes on repeating till it reaches to the termination codon. Okay, all the steps are repeated till it reaches to the ribosome reaches to the termination codon. At this stop codon, there are three termination codon we have studied, that is UAA, UAG and UGA. It is exposed at A site. These termination codons are exposed at A site. Okay, and it is not read. These termination codons are not read and joined by anticodon of any tRNA. So, what is going to happen here? The release factor binds to the stop codon. There is a release factor that binds to the stop codon. Okay. At stop codon, a protein called release factor binds to the A site and no tRNA for stop codon. Thus, no amino acid tRNA complex will be formed. Okay. And what is going to happen? The two subunits, finally, what is going to happen? The polypeptide chain will be released. Okay, polypeptide chain is now released in the cytoplasm because it has reached to the stop codon. Release factor is acting here. Stop codon comes here. Okay, mRNA is moving. Stop codon is coming. Because of the stop codon, release factor is working. And when release factor is working, it releases the polypeptide chain. And this pre-polypeptide chain is now released into the cytoplasm. And the two subunits are separated. The two subunits are separated from each other in the cytoplasm. Both are set free in the cytoplasm. Okay. Now, this completes the process of translation. Now, there in mRNA also has some additional sequence. You can see here mRNA, the stop codon has reached. But this mRNA has some additional sequence at both the ends, that is 5' end and 3' end. Okay, at both the ends they have some additional sequence that are not translated, and this are referred as untranslated region, that is UTRs. They are present at both the regions, that is 5' end and 3' end. UTR at the 5' end, 3' end. Okay, untranslated region. They are required for efficient translation process. And finally, mRNA is also released in the cytoplasm. It gets denatured by nucleases immediately. Hence, mRNA is short-lived. So here we complete this process of translation. Okay, thank you.